That's what I want everyone here to think.
All right, everybody, thank you for coming today. I have the pleasure to introduce Amber Johnston. Amber is a biological science technician with the USDA ARS Aquatic Animal Health Research Unit in Auburn, Alabama. Today, she'll be presenting some work that she completed during her master's with the Michigan State University Aquatic Animal Health Laboratory prior to moving down to Auburn. She's going to talk to us today about Lake Sturgeon herpes virus 2 and its virulence in juvenile Great Lakes lake sturgeon and susceptibility to multiple disinfectants in vitro. So Amber, the floor is yours.
Awesome. Thank you again. Thank you everyone for bearing with me during my technical difficulties, but we've all been there and had that happen at least once or twice at this point. So to get things started, I just want to acknowledge my co-authors. They've been a tremendous help for all this research throughout my master's. And I also want to acknowledge the meeting organizers for once again putting together an awesome opportunity for its students and early career folks to present their research. So without further ado, I'll be talking about some research today that I completed towards the end of my master's. And while I've since moved 900 miles south to work with the USDA in Auburn, Alabama, there was a couple more things of my master's thesis that I hadn't really had a chance to present to this audience yet. So I'm taking advantage of that opportunity today. And without further ado, I'll get things going. So the lake sturgeon is the only native sturgeon species in the Great Lakes where they're an ecologically and culturally important fish. And unfortunately, recent estimates suggest that their populations are less than 1% of historical abundances. And because of that, substantial resources have been allocated to the hatchery rearing of lake sturgeon for conservation purposes. So what exactly does hatchery rearing of these fish look like? So in the Great Lakes, resources are allocated to the propagation of juvenile lake sturgeon in rearing facilities with the intent of releasing them back into wild populations. And because there's no maintained adult lake sturgeon brood stock in the Great Lakes Basin, there are two main ways that this happens. First, wild adult lake sturgeon are captured where eggs and sperm are collected and fertilized artificially in the rearing facility. And from there, fish are hatched and raised for a few months until the risk of predation is a little bit lower and they're then released. Additionally, facilities are propagated via the collection of naturally spawned larvae directly from rivers and streams while these larvae are in their drift phase. So this is basically the phase of life where they're most vulnerable. So hatchery managers and personnel will intervene, get them out of those rivers and raise them until they're in a less vulnerable life stage. So while these are innovative techniques, these methods mean that whatever problems or pathogens facing wild adult lake sturgeon could also come to fruit in these gametes or larvae that we see in the facilities. And I just want everyone to hang on to that thought as we move forward. So while hatchery rearing in the basin has become a pretty common practice, it unfortunately isn't exempt to challenges. So many, if not all of these facilities basin-wide grapple with occasional sizable mortality events. And unfortunately, using routinely employed laboratory tests, the cause has sometimes gone unexplained. And further complicating that issue is that there really wasn't a whole bunch known at the time regarding the presence of infectious diseases in adult lake sturgeon populations, which, like I mentioned before, are oftentimes producing gametes or larvae to populate these facilities. So therein lied a pretty substantial unknown. So given that there was an apparently large knowledge gap to be filled in here, Dr. Locke and collaborators sought out funds to support a study that targeted this exact issue. So with that, I was afforded the opportunity to conduct a large-scale lake sturgeon infectious disease study funded by the United States Fish and Wildlife Service, Great Lakes Fish and Wildlife Restoration Act. And the whole goal of this study was really to just figure out what risk these emergent or endemic fish pathogens could be posing to Great Lakes lake sturgeon populations. So this study and associated findings made up just about the entirety of my master's research. And while I won't go into the nitty-gritty details of this large-scale disease surveillance study, to move forward with this talk, I need to highlight one key finding that formed the foundation of the research I'll be presenting to you all today. So if I've been so fortunate to have you in attendance of my previous talks, either here or other fish health section meetings, you probably have an idea of where this is about to go. And I just ask that you bear with me as I do my best to sum up 2 years worth of collaborative work. So while sampling adult lake sturgeon in the Great Lakes, I observed evidence of skin damage in the form of these opaque plaque-like lesions. And these lesions varied in intensity and also had a prevalence of about 1 to 14% across both sites and years when I sampled. So through reviewing the literature, I noted that these lesions were very similar to those induced by acid herpes viruses. So with that in mind, we pursued a pathway that focused on determining what this S-penciled herpes-like virus really was. So as it turns out, the causative agent behind those lesions was indeed similar to S-penciled herpes viruses, but was actually an entirely novel alloherpes virus. So much of the work done to isolate and characterize the novel Lake Sturgeon herpes virus 2 was published in November, and in this paper, we not only link Lake Sturgeon herpes virus 2 to gross lesions observed in spawning adults, but we also detailed the isolation of the virus, which was a critical step in pursuing further research. So now that I've hopefully given adequate context as to how this story all began, for the remainder of my talk, I'll be diving into some experiments conducted towards the end of my master's. Some are detailed in this manuscript, some of which I'm presenting here for the first time outside of my defense. So while there are a mountain of unknowns associated with the detection and isolation of a novel virus affecting one of the most intensively managed species in the Great Lakes, my project was only designed to be a master's, so we had to be a little bit selective and prioritize what unknowns we seemed were most important to address at the time. So like I mentioned previously, a key component of lake sturgeon conservation is the hatchery rearing of juvenile lake sturgeon, where larvae and gametes are collected from wild adults. And as it turned out, the virus was indeed found in a lake sturgeon population that is used to populate one of the largest rearing facilities in the Great Lakes Basin. So with that in mind, we wanted to figure out how this virus could behave in experimentally exposed juvenile Great Lakes lake sturgeon, especially given the propensity of some alloherpes viruses to be transmitted from adults to juveniles via vertical transmission. Given these facilities are populated with gametes or larvae from potentially infected adults, we wanted to investigate ways in which we could arm hatchery personnel with tools to prevent the potential spread of Lake Sturgeon herpes virus 2 in the hatchery environment, which I'll expand on a bit more later. So with that in mind, we developed two objectives. The first is what I'll start here with today, and that is to elucidate the virulence of novel Lake Sturgeon herpes virus 2 in juvenile Great Lakes lake sturgeon. So as many here are likely familiar, a pillar for linking a virus to disease causation is the fulfilling of Rivers' postulates, which requires pathogen recovery from an infected host. That virus then needs to be cultivated in vitro and inoculated into a naive host. And that naive host must then exhibit gross signs of disease, and the virus must be re-recovered from that infected host. So fulfilling this allows you to more directly link a specific virus to the manifestation of disease. So with that, I began a series of small-scale challenges to assess the effects of temperature, dose, and route of exposure. And this is all done with an eye towards developing an ideal experiment model, to begin fulfilling Rivers' postulates. So based on pilot challenges and because it most closely mimicked virus exposure in the wild, I pursued an immersion bath exposure experiment. However, this meant I would need to cultivate a large amount of virus to immerse multiple groups of fish. So while I didn't fully detail the process or story of isolating this virus here today, in brief, when I had first tried to recover this virus via cell culture, I was unsuccessful. But in my next go around trying to isolate this virus, I did succeed. However, it was on a very, very small scale. And this scale was so small that it actually had to grow up 1,000 times more what I had going in order to be able to conduct this challenge. And not only that, I had to simultaneously grow up enough cells to sustain it. And I'm sure there are a lot of people that work with viruses out there that can empathize with me here. But the cell line I was working with was a bit difficult to deal with at times. So this process alone took nearly as long as the entire experiment. But nonetheless, I did succeed and I was able to grow up enough virus to pursue this experiment. So for the next few slides, I'll have this little roadmap at the bottom aimed at guiding you through the multiple stages of this immersion bath trial. So now with quantified virus in hand, I immersed 6 replicate groups of 8 fish in a virus suspension for 1 hour before pouring them back into their respective tanks. Additionally, 6 groups of 8 fish were mock challenged with uninfected cells and media. So less than 2 weeks post-exposure, I began appreciating a few initial signs of disease, including decreased reactivity to feed, general lethargy when compared to negative controls, and perioral hemorrhaging, which you can see here. Let me get my laser pointer going. So disease continued to progress, and 19 days post-infection, this first mortality presented with more widespread hemorrhaging around the eyes, But what was most predominating here was this ulceration around the caudal peduncle, and this also extended deep into the musculature and resulted in near complete loss of the caudal fin. So this continued, and I also appreciated evidence of emaciation, as you can see here, but was most consistent across these mortalities was this ulceration of the caudal fin. And I want to stop here quick and address the fact that some of you in the audience might be thinking that this type of lesion is very resemblant of that noted when there's an infection with a columnaris causing bacteria. And we thought the same thing. So in addition to collecting tissues for virus re-isolation, we actually also cultured from these lesions on a media that would be selective or not selective, would allow for the growth of Flavobacteria species. And all of these cultures that we did were negative for any yellow pigmented bacteria. So we covered our base there. And like I said, the lesion development that we saw here was pretty consistent across all these replicates. As you can see throughout this challenge, we kept seeing the same ulceration of the caudal peduncle up until the very last mortality at 76 days post-exposure. And ultimately, this experiment was concluded at 112 days, so just under 4 months. So now what I'm showing you here is the cumulative percent mortality on the y-axis as a function of days post-exposure on the x-axis. And I'd first like to highlight that at no point during this nearly 4-month-long experiment did we see a mortality in any of our negative control replicates. In contrast, across the 6 replicates of virus-exposed fish, cumulative percent mortality reached near 35%, which when considered in the context of hatchery rearing, the possibility of losing over 1/3 of fish reared is pretty problematic. So as fish were worked up either during or after the study, like I briefly alluded to before, I collected a multitude of tissues for inoculation onto cells, because if you recall, to fulfill Rivers' postulates, I needed to re-recover the pathogen. And so it goes, once I inoculated tissues from caudal lesion samples onto cells, I did indeed find evidence of a replicating agent with cytopathic effects identical to that observed previously with the original virus these fish were infected with. And further analysis concluded that the virus I'd isolated and detected on cells was near identical to what fish were initially infected with, so we did indeed re-isolate Lake Sturgeon herpes virus 2 from these infected fish. So we had previously linked this alloherpes virus to the development of gross signs of disease in adults, as I mentioned in the very beginning. And now we had also found that it has the capacity to kill juvenile lake sturgeon or induce disease. So from there, we wanted to figure out how we could mitigate the possible negative effects of this virus in rearing facilities. So given the risk posed by Lake Sturgeon herpes virus 2 and our now ability to grow this virus and conduct in vitro experiments, we wanted to figure out what exactly this finding meant for those rearing lake sturgeon in the Great Lakes and how we could begin to help. So a pillar for disease prevention and control is through the upholding of biosecurity. And the use of disinfectants is an important component behind biosecurity that is used by those rearing fish in the Great Lakes. So disinfectants are used for a variety of purposes in aquaculture. These include surface disinfection of the eggs in an effort to prevent transmission of disease from parents to offspring, the treatment of disease in both eggs and post-hatch fish, and the disinfection of surfaces and equipment used under hatchery and field conditions. Products are approved for use in aquaculture or hatchery settings. I chose 3 approved for use hatchery disinfectants to see what effects they would have on Lake Sturgeon herpes virus 2, because we figured it best to just start with these tools that are already ready and being employed. So with that, I developed the second objective of this study, which was to assess the susceptibility of Lake Sturgeon herpes virus 2 to 3 routinely employed hatchery disinfectants. So to conduct this experiment, I started with a Lake Sturgeon herpes virus 2 isolate. I exposed that isolate to 3 different disinfectants, each at 2 different concentrations, which were chosen based off of either the guidelines for use, purposes for use, or previous experiments. Further, each disinfectant concentration was exposed for 3 time points, including 1 minute, 10 minutes, and 30 minutes. And this is all done in duplicate, but just after exposure to that time point, disinfectant was deactivated and virus was plated onto TCID 50 plates, which I recognize is a bit of a mouthful, but it's basically just a standardized method for virus quantification. So what were the results? So to determine how efficacious any particular disinfectant was, I compared the TCID 50 results of virus unexposed to disinfectants to the TCID50 results of virus that was exposed to each disinfectant at each concentration and time point. So all these values were calculated based on averaging the two replicates. And when we looked at Ovidine, percent reduction at 50 and 100 parts per million was over 99% after just 1 minute. And furthermore, after 10 and 30 minutes, percent reduction was nearly 100%. Ovidine is commonly used two ways that I highlighted here. And those two ways are 50 parts per million for 30 minutes or 100 parts per million for 10 minutes. So these in vitro results suggested that it's highly effective at deactivating Lake Sturgeon herpes virus 2. And while these findings were encouraging, it's important to note here that iodine only disinfects the surface of the egg. So if Lake Sturgeon herpes virus 2 were to be transmitted inside the egg, it may not be as effective. And furthermore, in vitro findings don't directly equate to how things would work in the hatchery. So in vivo hatchery studies are still warranted to test if this would really work in a hatchery environment. So moving on to hydrogen peroxide, which is commonly used to treat various diseases throughout different life stages. At 500 parts per million for 1 minute, the disinfectant only induced a 58% reduction. Fortunately, though, when employed at higher concentrations for longer time points, percent reduction increased to above 90%. And these results suggest that the longer this virus is exposed to peroxide, the higher percent reduction is likely to be. But similar to iodine, I just want to put this in here again now that how this translates to a hatchery environment remains to be determined. However, we do see a good amount of promise here with both of these disinfectants so far. So lastly, we have Virkon Aquatic, which was highly effective even after just 1 minute of exposure. So these results suggested that Virkon Aquatic is a very, very promising means to deactivating virus on surfaces or tools. And while like all other disinfectants that I've talked about so far, studies in a hatchery setting are necessary, these current results are very promising. So overall, while work is still needed to figure out exactly how this will look in a hatchery, my in vitro results showed that these 3 disinfectants show great promise in the deactivation of Lake Sturgeon herpes virus 2. So there are 2 key conclusions first coming out of this talk. And the first of those is by conducting a statistically relevant immersion challenge, we fulfilled Rivers' postulates and definitively determined that this virus is indeed capable of inducing mortality in juvenile lake sturgeon, which begs the question, has this virus been playing a role in unexplained mortality events that we've been seeing in these facilities? Or perhaps has this virus been hanging out in the background, also contributing to long-standing lake sturgeon population declines? And while the experimental conditions I used here might not be directly applicable to hatchery environments, I just want to highlight that should they be like even kind of applicable to the hatchery or translatable to hatchery environments, if this virus were to end up in a facility, if you're facing losses of 35% or worse, and you're talking about some of the most intensively managed fish in the Great Lakes Basin in terms of both time and resources, that's a pretty big cause of concern and definitely warrants further understanding. Fortunately, though, not all is bad because these in vitro studies showed that routinely used disinfectants can be efficacious under in vitro settings. So I know it can be tempting to think, perfect, it works, it can kill the virus 100%, we're good to go. In vivo studies are definitely still needed to see how these would truly work in a hatchery setting. Regardless, though, these 3 disinfectants all seem to so far be promising means of disease prevention and control. Ultimately, the research I've conducted here is positioning us to not only address how we could prevent and control the spread of this virus in rearing facilities, but will also serve as the pillars for further understanding of the novel Lake Sturgeon herpes virus 2. So with that, I'd like to acknowledge the United States Fish and Wildlife Service for funding all of this research, the Janice Lee Fenske Excellence in Fisheries Management Fellowship, which provided me a lot of funding throughout my master's, Drs. Matt Griffin, Dr. Bradley Richardson, and Dr. Jeffrey Waldwieser, the Black River Sturgeon Facility crew, Michigan DNR, current and past MSU Aquatic Animal Health Lab members, as well as my former master's thesis advisor, Dr. Tom Locke. So my contact information is there if anyone has any questions. And despite the technical difficulties, I do think we still have plenty of time for questions. So I'll take them all.
Excellent. Thanks, Amber. Fantastic talk as always. If you have a question, feel free to chime in, unmute yourself and ask, and or place it in the chat, and we'll go from there. So Meg, do you have a question? Or was that an applause? Oh. Emily Churchman.
Hi, Amber. Great talk, by the way. I was wondering if you happen to see or know or hypothesize any sort of viral virulence factors that might have led to those lesions on the caudal fin.
Yeah, so that's an awesome question. I might not have the perfect answer, but I'll give it my best shot. So a lot of herpes viruses tend to have an affinity for like epithelial tissues like the skin. And like I mentioned, we had seen lesions that had those like that kind of like hyperplasia of the skin where we saw those opaque like plaque developments. And sometimes when I would see those lesions in the field, they would eventually develop into ulcers. So you would see that plaque and then you would start to see hemorrhaging around the margins, and you would sometimes see ulcerations associated with those plaques. So while I didn't necessarily see plaque development in these juveniles, I wasn't entirely surprised to see that this virus really had an affinity for the skin, like a lot of other herpes viruses do. So I don't know exactly virulence factors, and I'm not sure if I totally addressed your question, but it was a little bit expected with those lesions. But like I mentioned, they were very consistent. Like every mortality I had in that study had manifested that type of lesion, and we did re-recover virus from it. So thank you.
No, that was great. Thank you.
We have a question in the chat. As a majority of herpes virus becomes latent in their hosts, do you know does this virus latently infect sturgeon?
That's an awesome question. And like I said, this was all pretty new. I mean, this work had just started about 4 years ago, and there's still so much to figure out here. Like latency is a huge component here that could be an issue that we don't even know about yet. So far, when I was working with this, we had only isolated virus or detected virus directly in those lesion tissues. So we hadn't really gotten as far as figuring out latency because we were really only looking at those fish that were like outwardly infected, and that's where we were finding virus. So it's a great question. I don't have a good answer for it yet, but it's definitely an awesome area for research. And it's my understanding that I do think research is going to keep going with this up at the lab at MSU. So I'm excited to see what comes out of it and hopefully stay a little tangentially involved. But yeah, I don't have a good answer for that, but lots work to do. So.
Do we have any additional questions for Amber? Well, she's here and on the line. Well, if not, we've hit 12:30, and just because there was only one participant, that's not always fair to just keep Amber on the hook under the interrogation longer. So if nobody has any more questions, we will adjourn for the day. A big round of applause, Amber. Fantastic presentation as always, and thanks for contributing, and thank you all for coming. It's great to see